this is Mrs. Murdoch, and we have a mystery to solve. I have some colored solutions here that I've put food coloring in, and each one of them has a different concentration of sucrose in them. They're just simple sugar solutions with some food coloring added. And the highest concentration of sucrose is 1.0 molar. It's really, really high concentration of sugar. And then the lowest has nothing in it at all, it's 0 molar, and they're all gradations in between. And I know which color is which concentration, but I want you to figure it out based on the data we're about to collect together. So what we are going to do is we're going to use something called dialysis tubing to make little chambers for each solution, which I've already done here on the table. If you look here, I've placed some of each color of solution into a separate little dialysis tubing bag. And I just want to introduce you to dialysis tubing just for a minute. It doesn't look like much, does it? It looks like a flat piece of plastic that doesn't look like a tube at all. But what I did is I cut it up into pieces and I soaked it for a while. So it's been soaking here for a couple hours. And when you soak it, then you can tease it open. If you rub it back and forth, it actually becomes a tube all the way through. And if you knot one end, and then you can pour liquid in using a funnel until you have some liquid in there, and then you knot the top end, you basically have a sealed chamber. So let me tell you a little bit about dialysis tubing. Dialysis tubing is a man-made polymer that has tiny little holes in it. It's that simple. But it has a very special use. You may recall that the word dialysis is a process that some people have to go through if they have damaged kidneys. Now, the kidney, when it's working normally for a person, is a filter. It filters all of the waste and toxins out of the bloodstream. So they're an incredibly important pair of organs, and if they're not working right, whether a person has some disease in the kidney or some diabetes can sometimes do damage to the kidneys. There's all kinds of reasons why your kidney might not work. But if, in fact, you need to go to have dialysis done, this tubing is involved in that. So, and it's a concentration manipulation question. So if a person is going in for dialysis, they have to go to a clinic, you sit down in a chair, and you have your blood drawn out of your body, and it goes through a machine, and it goes through inside this tubing. And then the machine has fluids around the tubing that have a different concentration, a low concentration of things. So then you know from diffusion that things will always move from where they're more concentrated to where they're less concentrated. So it's basically an artificial filter for a person's bloodstream. It will draw out all of the nitrogenous wastes and toxins from the blood, and then the clean blood goes back into the person's body. So this is a perfect thing to talk about for this unit, which we're talking about cell transport and things moving across membranes. And in fact, this is a process that is taken advantage of in whoever built the first dialysis machine. They really saved a lot of lives because there are a lot of people who need that. So that's what dialysis tubing is. Here are some dialysis tubing chambers that contain these different amounts, these mystery amounts of sucrose solutions. Now, if you watched my video before, we talked about the sizes of molecules. You learned about different types of carbohydrates in your last unit. So you should remember, think in your mind, how big is sucrose? And is it going to be able to move through the holes of the tubing? And how are we really going to figure out here what these mystery things are? So what we're going to do is right now we're just doing the very first part of the lab, and then we're going to do another video that shows the results. So the first thing we're going to do is we're going to take an initial mass using an electronic scale of each of these bags. So I'm going to take the purple one first. I'm going to bring it over to the scale here. It is tiered. And my assistant get a really close look at that. Okay, good. So that's my initial mass, and then I'm going to immerse it in some tap water. There. Okay, that's purple. All right, here's the next one. I'm going to try to do this quickly because I want them all to go in around the same time. Okay. And immerse it. Right. Here's a clear solution. Okay. And immerse it. Here's the orange solution. Okay. And I'm gonna immerse it. Here's the yellow one. Okay. And immerse it. Here's the green one, last one. Okay. And immerse it. And in another 30 minutes, we will talk again. Thank you.